Dear class, unfortunately, lecture capture today didn't work. They had a system-wide failure. So I'm just going to give this lecture and post this on YouTube, and I'll announce it for you. We never did this, but I thought I'd do it for you just to help you out. So it asked common ancestor of fungi and animals. Where would that be? That's A. Well, if we go up to fungi, the animals, we see they're very closely related in terms of overall phylogeny. They share a node right there. And so A would go right there. Okay. B, common ancestor of archaea and eukarya. Okay. So let's look at the node again for a common ancestor. It's right there. And so what we would look for, common ancestor for archaea, that would be B. And then common ancestor of all life, that's pretty simple. Again, the node. And common ancestor of plant, fungi, and animals. Again, the node. And then common ancestor of eukarya. Okay, good. Okay, onwards and upwards. So, in terms of the diversification of life and the origin, we don't really know very much. Now, I mentioned early on about the tree of life and how it had one trunk, indicating there was one origin, and then from there all other life branched off from that during speciation events. Now that's different than if there was separate origins for different groups of organisms. And I asked what the evidence was for one or the other. Well, if it was multiple origins, you would expect there wouldn't be the kinds of similarities that all life would share compared to multiple origins. So we know that there is only one DNA code, and the code is read the same way in terms of transcribing and translating into proteins. There are not other genetic codes. Furthermore, there are ribosomal RNA, which is what is then finally translated into proteins, is shared by all life as well. So we have very good evidence only for a single origin. Now, there could have been other origins of life that, but that didn't persist and have gone extinct. But we haven't found any evidence of that either, even though we have stuff as old as 3.7 billion years for cyanobacteria. So that's the state of affairs. We don't know how life started. You can read in the book about some various sorts of experiments that people have done, but of course no one really knows, and it's probably one of those unanswerable things. The big question, of course, is life rare or is it common in the universe? And the universe is very, very big. If you look at how big a galaxy is and how many possible worlds with planets that might harbor life, it's probably a high chance that there's going to be other examples of life in the universe. But we don't know. So the basic unit of life, of course, is a cell. And a cell generally has a plasma membrane. This is a bacteria, so it has a cell wall. All things have a outer membrane. I don't know about a capsule. We definitely have DNA, right? This is the cytoplasm containing ribosomes. All life has that. And then there's a little circular piece of DNA right here called a plasmid that codes for certain things, and it can be transferred to another bacteria. And then, of course, finally, there's pili, and there's a flagellum. Not all bacteria have flagellum. Okay, only some do, and it's a very useful way to actually travel, but as you'll see, there's a great diversity of bacteria. So the basic unit of life is a cell with DNA, ordered structures, regulation of internal conditions, homeostasis, growth and development, energy use, response to stimuli, and of course reproduction and evolution. Now, there's a question about whether, like I asked, is there anything else that might be considered life? And we came up with the viruses. And, of course, viruses have DNA, or they have single-stranded RNA, but they're both use the same kind of translation in terms of translating into proteins. There's a little bit of a difference, but all life uses that. So life with cells uses RNA for part of the translation machinery. Okay, but viruses in some cases use that also as the genetic code as well. Okay, so there's 3 domains of life, and but we're dealing now with a group called prokaryotes. These organisms include bacteria and archaea. Okay, and prokaryotes don't have a nucleus or any organelles. So they're very simple organisms, but they are a cell. Whereas eukarya are organisms like our cells, but also unicellular, but they do have a nucleus and they do have other organelles like mitochondria. Okay, so if we were just going to use this one character, which is whether you have organelles and a nucleus or not, we would probably group this group together. And the tree would look like this. Where we have eukarya with organelles, bacteria without, and archaea without. But that's not what we actually see when we look at the 3 domains of life. We actually see that, in fact, archaea and eukarya share a most recent common ancestor. Then either of them share with bacteria, which happened earlier. Okay, so now that's kind of strange. Why would archaea not have a nucleus, and eukarya would, and other things? But it appears that eukarya is actually, so eukarya is a combination actually of archaea and bacteria. Inside some archaeal cell, there are organelles, and one lineage, mitochondria, comes from protobacteria, as we'll see, and the other, chloroplasts, come from cyanobacteria. And then, we're not going to get into this, but as far as the nucleus, it might be actually viral in origin. So these are mutualisms that evolved over time from free-living organisms. Okay, so our tree, what would you call this tree, by the way, in terms of what kind of grouping is this? Is it monophyletic? Is it polyphyletic? Or is it paraphyletic? And I mentioned, because we only have these 2 groups, and not all of them. If it was just archaea and bacteria based on the nucleus not having one, then that would actually be what's called a paraphyletic group. Because, remember, it includes some but not all descendants of a common ancestor. Okay. Actually, okay, so here we are again. Now, there's also another thing that's in common with all of them, all life, and that is enzymes used for DNA translation and transcription, and we're talking here enzymes and RNA, okay? And part of translation is ribosomal RNA, which then turns messenger RNA into proteins. The thing is, it turns out that ribosomal RNA for eukarya and archaea is actually more similar to each other than either of those for the RNA that bacteria uses. So, here we have a divergence in characters. Prokaryotes don't have a nucleus, but in terms of RNA and ribosomal RNA, eukarya and archaea share more recent type or similar ribosomes. Okay, so that suggests that it's actually archaea and eukarya that are in fact more closely related to each other. And there's a whole bunch of other things. So also archaea is a new, it was found in the last 40 years by Carl Woese. Before that, people didn't even know about it. Okay, and then we went over this. Prokaryotes are monophyletic. It's false. If it was like this, then archaea would be monophyletic because eukarya would be on its own branch, but it's not. And then archaea are sister clades because they share most recent common ancestor. Okay, now prokaryotes. So really, a prokaryotes is really a bad name for this group because it's paraphyletic, and we try to stay away from paraphyletic groups because it doesn't actually show evolutionary history and when branches happened relative to others. So we will use prokaryotes, but with the understanding that it's made up of bacteria and archaea. Most of these are unicellular. Some of them are somewhat colonial, and some make biofilms. Okay, but both lack a nuclear membrane. In other words, they lack a nucleus. Okay, very important. But there are some differences between bacteria and archaea, as I mentioned. And then you can see in eukaryotes exactly, there's a nucleus, there is mitochondria. Doesn't show chloroplast here because it's not a plant cell. Okay, so prokaryotes are hyperdiverse. It turns out that they're very small in size. There's over 400 species in your small intestine, 1,000 species in your large intestine, 128 species in your stomach lining, 700 species in your mouth. In fact, we have bacteria and archaea and fungi and viruses all over and inside our body, and they actually don't make us, they're not pathogens necessarily. They're commensals, or in some cases, they actually provide a benefit, like some bacteria in our gut actually makes important vitamins that we need. Also, in terms of overall biomass, prokaryotes, or microbial life, but prokaryotes make up a tremendous amount of total biomass on the Earth. And they're found everywhere from up in the atmosphere all the way deep underground and inside rocks. So bacteria and archaea, they differ though in structure, physiology, biochemistry, as I've alluded to. Now, as I mentioned, prokaryotes, archaea, and bacteria, they live everywhere, and some of them are what are called extremophiles. They live in very, very extreme, and one would say difficult environments, and yet they appear to thrive in some of those. So, up on top we have Yellowstone National Park. We have this little pond here with different kinds of bacteria, different colors, cyanobacteria, also perhaps some algae, I'm not sure. And then you can see the same sort of thing in this fumarole. You can even find bacteria or archaea living almost in the inside a volcano. And in fact, you can find them at these big smoker vents in the ocean that pump out tremendous amounts of minerals and stuff, and that some of these bacteria or archaea actually use that as energy, like photosynthesis, but it's chemosynthesis. So these extremophiles include too hot, too cold, at least for us, too acidic, too basic, too salty, all that. Okay, now, most people think of bacteria that cause infection, right? And they do cause infection. A lot of them actually do. We have a whole list of them, and different areas of the body that are attacked by bacteria. But it turns out that most bacteria in our environment we either don't interact with at all, or if we do, they're actually commensal. In other words, we've got tons of bacteria in our gut and tons on our skin and everywhere else, and we live together, and they might get a little bit of something from us, like a good place to live. Some of them actually provide things for us, like the skin on our the bacteria on our skin actually protects us against other bacteria that might cause infection. So they're almost part of our immune system. Okay, so only some bacteria. Now, some bacteria also are infectious, but they're not pathogens. In other words, they can infect cells but not cause any disease. So that's also important. And furthermore, it doesn't mean that bacteria, the same bacteria, are always commensal or always pathogenic. We found this out mainly from HIV, because HIV disables the immune system, and people were actually dying of these opportunistic infections caused by bacteria, caused by fungi, caused by eukaryotic single-cell parasites, and also cancer. And so we learned a great deal about the immune system because of HIV. And these opportunistic infections, these are organisms that normally don't cause us any problems whatsoever. Our immune system, they live with us, but our immune system keeps them under control. And it's only when the immune system was removed from by HIV that all of a sudden they sprang into action and caused disease. And I mentioned also this antelope in Eurasia, Sega antelope, and recently there was a big die-off. and it seems to be associated with very warm weather and high humidity, and it decimated the herds of saga antelope. You'd have a herd of, let's say, 300. It was during calving season, and every single one of them died. They were wondering, what it might be, and they recovered a bacteria that actually is commensal with them. In other words, it lives with saga, never causes any disease with saga, but it's only at these periods of times, and then for some reason, these conditions cause some sort of mechanism where it gets into the blood, septicemia, toxic shock, and it basically kills them all. So it's not like you're always stuck with a disease. Also, as you know, some people have stronger immune systems than others. Now, bacteria, we've also used them for a number of different things. More importantly, besides, here's water pollution and stuff. But we also use them like bacteria to make drugs, like insulin. We make huge vats of human insulin by putting a human insulin gene, into a bacterial genome, and it produces insulin for us. Now, something that's kind of interesting, the only organisms that we really know about are organisms that we can recover and stain and perhaps grow, culture, like some bacteria we can culture, some archaea we can culture, not very many, obviously some different kinds of fungi we can culture, but there's a lot more out there where we just don't have the right medium or conditions to actually grow those things. And until recently, we didn't even know they existed. But they do. And we know this because now we have DNA sequencing, and people have been collecting samples from all over the world and then sequencing the DNA without knowing about which organisms are in there. And then you use the DNA to see who's more closely related to each other in terms of various kinds of bacteria, archaea, whatever. And we know there's all these kinds of microorganisms that we've never seen, we didn't even know existed, but we do now because we've collected their DNA and we've sequenced it, and we see in a phylogenetic tree where they actually all belong. It's called metagenomics. Prokaryotic basic biology. They have a cell wall covered by a sticky capsule. Bacteria have a peptidodiclan in their cell wall. Archaea don't. They have a unique phospholipid, which is a phosphorus and a fat in their plasma membrane. So there's some very basic differences between bacteria and archaea. And peptidodiclan is a single polymer that forms a net-like mesh around the cell. You can actually see it. Okay. And this is the cell wall, peptidodiclan. Okay. Now, so just so you remember, antibiotics only work on bacteria, and only on some bacteria. Mostly they work by degrading the cell wall of bacteria or preventing the synthesis of the cell wall. Okay, that's how they work. And it interferes with peptidoglycan, there we go, synthesis. Now, so here it kind of asks, remember, basically antibiotics, in this case penicillin, kill bacteria. It's like they're a toxin, okay? So if antibiotics such as penicillin interfere with peptidoglycan synthesis, do you think that human cells have peptidoglycan? So let's think about this. If our cells were made of this substance, this protein, do you think penicillin would be harmful or toxic to our cells? Yes. But in fact, we can take penicillin with no effects whatsoever. So it's pretty obvious that bacteria have peptidoglycan in their cells as part of their structure, but we do not. So prokaryotes, archaea, and bacteria are a diverse lot, and they vary in size from very small, 1 micrometer, 0.5 micrometers, probably 200-300 micrometers, right? And they also vary in shape. So on the left we've got cocci and spherical bacteria. In the middle we have rods and bacilli. On the right, spiral helix like spirochetes. One of the common ones is one that causes syphilis and Lyme disease. Okay, so prokaryotes are very diverse and they're found everywhere. So motility in terms of bacteria, in other words, how they move, varies. Some don't have anything to help them move whatsoever. Others have flagellum. I'm not sure about other methods of motility, but some actually are actually immotile. In other words, they don't move. In fact, they have a lot of them make biofilms, and they stick together so they don't move. Okay, so this is redraw with your 3 taxa: bacteria, archaea, eukarya, and label as a sister group. Okay, so here we go. I'll do this for you. So we've got, we'll start from the bottom. We'll start with bacteria, and we'll start then here and here. And here we'll have bacteria. Here we'll have archaea. Here eukarya. They share a node here. Whose sister? Archaea and eukarya. And there's also another sister group, bacteria plus archaea, eukarya. Okay. So when did life originate on Earth? You can read about this in chapter 26. We don't, the first evidence for life, as I mentioned earlier, was about 3.7 billion years ago. We know this, looks like it was mainly something like cyanobacteria that made these kinds of structures. So not that long, the Earth formed 4.5 billion years ago, so that's 0.8 of a billion years, or 800 million years. Now there's probably, in fact, life, some sort of life before then that didn't leave any residue or anything like that. Also, cyanobacteria use, they do photosynthesis. In other words, they use light to turn light and carbon dioxide into sugars, carbohydrates. But it's likely prior to 3.7, in fact, probably 4 billion years and older, the Earth was getting bombarded by all sorts of things like asteroids and comets and all that. It was a very violent early, when the solar system was being formed. And there's a good chance that, in fact, life might have started in some primordial soup, perhaps underwater, but near, let's say, a volcanic vent or something with lots of minerals and stuff. And they, instead of using photosynthesis, they actually use chemosynthesis, where they used some other reducing agent, oxidation agent, instead of oxygen. Now, and then 3.7 billion years ago, we had all these, there were probably other kinds of bacteria that weren't photosynthetic, right? So here's a bacterial phylogeny. On the left, we've got, it's kind of, they really shouldn't say thermophiles. They're not all related to each other. There's a gram-positive, which is one major branch. There's another major branch composed of spirochetes and cyanobacteria, and then there's proteobacteria. And with this metagenomic stuff, we might find some other stuff that we didn't even know thought existed. So we'll first talk about proteobacteria. They are one group, as we can see, of bacteria, right? And they include all sorts of different kinds of bacteria, some that cause disease, that are pathogens, others that aren't. Interestingly, they're the group that probably gave rise to mitochondria in DNA. So let's take a look at this from a perspective. Okay, so we have these mitochondrial DNA. And mitochondria, by the way, have their own DNA, as I mentioned. Okay, now some of it has been transferred to the nucleus, but we won't get into that. Okay, proteobacteria also are involved in their general. There's a big lineage of bacteria. Most bacteria in general don't cause disease in humans, but some do. We've got some right here. E. coli, some branches of E. coli cause problems. Salmonella, which includes typhoid fever. Vibrio, which includes cholera. And then H. pylori, which is a proteobacteria involved in the basis for ulcers. We have a cell and an archaea cell, an early archaea cell, and then we have a tiny little bacteria. Apparently what happened was the large cell came along and then engulfed small cell, the bacteria. Or perhaps the large cell was invaded by the bacteria, the proteobacteria. Now, we know there's some evidence that to show that mitochondria are in fact similar to proteobacteria. First of all, they have small circular DNA, just like a proteobacteria or a lot of bacteria. They have a cell wall made up of the same material that proteobacteria do. And let's see, they reproduce by fission the same way that bacteria do as well. And so there's a lot of evidence that this is kind of what happened, perhaps 1.4 to 2 billion years ago. And that gave rise to mitochondria, which are central to aerobic respiration, because proteobacteria, in fact, do aerobic respiration. They need oxygen in terms of taking carbohydrates and breaking them down, glucose metabolism. And it's the most efficient sort of metabolism, and it generates the most ATP, which cells use to do cellular work. Okay, so there's a lot of evidence that this is exactly what happened. Okay, so, furthermore, one other thing. The flagellum also appears to have originated from bacteria as well. Okay, so here we are with the biggest group of bacteria. It turns out it was thought that stomach ulcers are caused by stress. And it was based on the assumption that the stomach was too inhospitable for bacteria to survive in, and so they couldn't actually be responsible for ulcers. This was an assumption, though, and actually not tested. Well, there was a microbiologist and a physician in Australia, and he thought otherwise. So, and he collected bacteria. One of them was Helicobacter pylori, and from the stomach. And so he thought, I bet you, he hypothesized that these bacteria were actually responsible for some stomach ulcers. So he couldn't very well do an experiment on other people by giving them, a nice cocktail of H. pylori. So he decided to make a concoction, drink it himself, and immediately gave himself gastritis, precursor to ulcers. And eventually he got a Nobel Prize, by the way. And definitively showed that a lot of stomach ulcers, not all, but a lot, are actually caused by this kind of bacteria. So when he did this, he had there's gastric ulcers in the stomach, and duodenal ulcers, which is the area, the lower stomach that leads into the small intestine. And he took total 22 samples and from the stomach, from a gastric ulcer, and 77% showed this bacteria. And then duodenal ulcers, he collected 13 samples, every one, 100%, all had this bacteria. So the fact that, first of all, he disproved the assumption that bacteria couldn't survive in the stomach, that was the first thing, and then actually demonstrating that these could cause disease. Yes, bacteria actually, they communicate with other bacteria, with other cells. They do this mainly through chemotaxis. That means, in other words, chemically, with organic compounds and various things. Vibrio is a bacteria, proteobacteria that causes, one of them causes cholera, which is a really terrible disease. It used to kill lots of people because their stomach contents would be evacuated, their intestines, all that, and they would become dehydrated, and that's what they die from, is dehydration. So cholera actually comes from, seems like there's the general place where it hangs out is oftentimes in the ocean, in shellfish. Shellfish seem to be a perfect habitat for them to hang out. And also inside the guts of some fish. And people who eat seafood, especially in the warmer summer months, often it starts a cholera epidemic. Now, most cholera, it turns out, though, actually comes from the continent of India, and it causes these outbreaks that have been traced all over the world. Salmonella is another proteobacteria. It too infects the digestive system. There's a number of different species. One, salmonella, general salmonella, doesn't, I mean, it's lousy to get, but it won't kill you. But another, typhoid fever, is a very serious disease that used to kill lots and lots of people. It reproduces by binary fission. It has all these different flagella, and its main thing is it's heavily involved in foodborne illnesses. Okay, now cyanobacteria. There are other organelles that are found in eukaryotic cells, and one of those is the chloroplast. And again, there's this woman named Lynn Margulis who proposed this endosymbiotic theory based on some earlier work by a couple of Russians at the turn of the 20th century. And she proposed that, in fact, eukaryotes that photosynthesize, including plants, algae, things like that, actually got the ability, by ingesting cyanobacteria, that then became part of the cell in an endosymbiotic process. Okay, so here we are. Okay, there's cyanobacteria. And again, there are similarities between cyanobacteria and chloroplasts in terms of their structure. They also have a membrane, of course, that looks very much like a chloroplast, though chloroplasts in plants have a really strong cell wall. So cyanobacteria appear to be responsible for chloroplasts in plants and algae. And it looks like it was a secondary endosymbiont, so it happened on a number of occasions. But first of all, there was first primary into like with a mitochondria, and then it looks like those cells that had the cyanobacteria were then engulfed again. Cyanobacteria are responsible for much of the oxygen in the atmosphere. They are mainly found in water, and either in marine or freshwater environments, and they're ubiquitous. Hold on. Also, they're also responsible in a lot of organisms for fixing nitrogen. Now, nitrogen is makes up, I think, I don't know, 70-something% of the atmosphere. It's not highly reactive, but all life needs it. It's the basis for DNA, for lots of proteins. And so the only way, though, free nitrogen that's found in the atmosphere cannot be fixed, cannot be utilized by most organisms. So it has to be fixed into a form, like nitrates and nitrites, that can be used by life on Earth. And some of that fixing is a result of cyanobacteria are able to take that nitrogen and turn it into a form. There are other bacteria that do that. There are some fungi that do that in conjunction as a symbiotic thing with plants. And then there's some plants that, because of their tight symbiotic interaction, are nitrogen-fixing plants like legumes. And so nitrogen is depleted from the soil, so oftentimes people will plant legumes to replenish nitrogen in the soil. So cyanobacteria can be very toxic in the chemicals they actually release as waste products. Here's the Green Revolution, of course. Now, no free molecular oxygen for the first 2.3 billion years of history. It looks like cyanobacteria were responsible for actually producing free oxygen. Now, it didn't show up in the atmosphere until there was an oxygen sink where it combined with all sorts of other minerals like iron and numerous things. Oxygen is highly reactive. And it's, but once it got to a certain point where all that was filled up, then there was free oxygen released into the atmosphere. Before that, there was none. And initially, that had a bad effect on microbial life because a lot of oxygen actually is highly reactive and toxic. So before cyanobacteria, there was anaerobic respiration using the fermentation process, which is much less efficient than photosynthesis is. And aerobic respiration, aerobic respiration, which is what mitochondria do. Okay, so they were the basis for that. Changed Earth's atmosphere and then finally gave rise to multicellular organisms. Cyanobacteria can convert nitrogen, as I mentioned, into organic form, ammonia or nitrate, nucleic acids and proteins. And here we can see what they look like. Okay. Spirochetes. So spirochetes are also a major group of long-coiled cells that stain gram-negative. A lot of them produce, some are pathogenic, like those that produce syphilis and Lyme disease, such as that. They're ubiquitous. And it looks like they're actually modified flagella We can keep going on that. Okay. There are also gram-positive bacteria, like these rod-shaped bacteria. Gram-positive bacteria, you use a red dye as applied. Gram-positive, dark purple masks the red dye. And gram-negative, the red dye stains the colorless cells. So these are different kinds of gram-positive bacteria, and then gram-negative. Gram-positive bacteria include anthrax, a lot of staph infections. Not all are harmful, of course. Most bacteria are. Thermophiles are, well, that's actually a wide range of different kinds of bacteria. They're not all one lineage, I don't believe. But there are a lot of bacteria that actually live in very extreme environments, like here in Yellowstone, in this very, very hot water, over 200 degrees Fahrenheit. In fact, some of them live in up to like 500 degrees. It's very, very, I mean, just normally you couldn't live there, but they do. It's possible that in terms of a lot of thermophiles actually use chemosynthesis instead of photosynthesis to generate carbohydrates. And it's thought, like in medioceanic rifts, there's these big volcanic vents that lots of minerals come out. They support a great deal of different kinds of bacteria, and then a whole ecosystem builds on that, including giant tube worms. It's thought that perhaps the first life on Earth that was able to generate food for itself, which would have been the basis for the food chain, came from these thermophilic bacteria in these vents, because the surface of the Earth from about 4 billion years ago, all the way back to the beginning of the solar system, 4.5, was almost too inhospitable for life to have evolved. But there were oceans and water bodies and things like that. There was lots of volcanism and various kinds of things, and it's thought that perhaps the earliest prokaryotes survived and were able to use chemosynthesis. Okay, so here's our cladogram. Again, we can see that archaea and eukarya are more closely related to each other than they are to other bacteria. As you saw here. Okay. And even though archaea doesn't have a nucleus, it's more closely related to. Okay. And then finally, there's this other group that we talked about. We've been concentrating on bacteria, but there's also archaea, the other domain of life, eukaryotic life. And archaea are ubiquitous. They were first discovered in the 1970s. In this case, bright color produced by hot sulfur-rich waters by oxidative activity of archaeans that convert in H2S to elemental sulfur. So, I asked you, which taxon is responsible for oxygen on Earth? Cyanobacteria. Which taxon is responsible for mitochondria on Earth? Proteobacteria. In eukaryotes. Okay. And that's it.